come to maybe back to basics, I should say, because strategies to combat antimicrobial resistance do not rest only with you and me. Right in the beginning, we heard that we need to take the administrators along with us. We need the backup of the policy makers, that is the government. And as we will see here, we will see that it is more to do with many other players which you and I cannot control. We can control what is in our hands. So we understand the different aspects of antimicrobial resistance, or what I call as the drivers of AMRs. What is the situation unique to our low-income countries, especially in the Southeast Asian region? How can all this be changed? Who all needs to act? And what are the alternative strategies to antibiotics? Does the future hold any promises other than the newer antibiotics which cost so much? So this is a beautiful slide. Where's that mic on? Can I have that hand mic, please? This is relative contribution of factors as a driver for antimicrobial resistance. And this is how much evidence we have in terms of literature. And the size of it will tell you how much it is contributing. So we only talked of human antimicrobial misuse, that is the purple which is highlighted here, high contribution and high evidence, right? But here we have animal antimicrobial misuse. So how many of you are aware that the poultry industry and the meat industry uses antibiotics liberally? Colistin is being used very, very liberally, and many other of the cephalosporins are used because they increase the content of meat. So if you want an animal which is to be sacrificed for meat, for human consumption, to become fat very fast, you give that animal ciproflox or a third generation cephalosporin, and that animal puts on lots of muscle very fast. So there is huge amount of it used in the animal industry. Then there was a time when lots of antibiotics were used. There was overproduction. This is somewhere in the 50s, 60s. And then there was so much excess that they were just rapidly thrown out into the environment. And even today, all the plants that are manufacturing antibiotics, their effluents are going into our rivers and all our water bodies and into the soil also. So what happens as a result of that? There is a lot of environmental contamination. Apart from that, a person who takes antibiotics, when that person, the fecal matter from that person would still have that antibiotic. So that also is contaminating our environment. And we know that water contamination in India is a big problem. So environmental contribution, you can see that there is moderate amount of evidence and it is a moderate contributor. Healthcare transmission, of course, we do not clean our hands as often as we are supposed to. And as a result of that, we have a lot of cross transmission in the healthcare. Interestingly, suboptimal rapid diagnostics. We do not have rapid diagnostics. All the tests that were talked about, how many of you have access to these tests? Let me have a show of hands. Dr. Kirti talked of a lot of these high-end tests like Carba-R. Have any of you used it? No. So only one person has used it here in the audience. So it is not that easy. We do not have that great rapid diagnostics. Vaccination is highly suboptimal. How many of you take influenza vaccine every year? You take it to protect yourself. Do you also tell your patients to take it? Yes. How many of those who treat COPD patients ask them to take pneumococcal vaccines? Are you a pulmonologist? Yes. So that's why he does. But mostly, we as intensivists, if a patient comes to us, we forget. At the time of discharge, we just say, go to pulmonary medicine for follow-up. But we don't know whether he's taking a vaccine or not. That should come up front. Why? Because it will prevent further episodes of such serious infections. And that is one of the ways of, if you do not have an infection, you do not have to give antibiotics. So you're preventing antimicrobial resistance. Suboptimal dosing, right? It's all very nice. Modi ji has come up with all these Jan Aushadhalaya, which gives the same drugs at a much cheaper rate. But do we actually know that these drugs work? We don't know. I know that at some stage, CMC Vellore used to test every new brand, put it through their micro lab and see if it worked. I hope they still do it. That's perhaps one of the best ways of testing it. Travel. Today what you have, see the way the NDM moved from Delhi right up to across the globe, and India protested so vehemently. It's not only India's problem, it is a global problem. And mass drug administration, rarely we have to give mass drugs. So all these have to be taken care of if we want to tackle antimicrobial resistance. Unfortunately, 99% of the things are not in our hands. But still, we can do a lot and do our bit properly by trying to be as best as possible when we work in a hospital setup. So what is the situation that is peculiar in India and other developing countries? This is from 2017 article in BMJ. The WHO Southeast Asia region has unique characteristics that contribute to the likelihood of increasing resistance to antibiotics developing in the region. So, what is it that we are contributing? So, this is a graphical, they called it an infographic, right? So, humans, in the community setting, there is human-to-human transmission. What is the level of risk? High. Human-to-human transmission in the hospital setting is obviously much higher. And antibiotics stewardship is low across the region. Antibiotics are widely available without prescriptions and are inappropriately prescribed. So directly pointing to you and me that we are not doing our job properly, right? That's what we all say. How is it setting in the community? Access to water and soap in the household can be very limited. Combined with poor knowledge and education about hygiene, transmission of antibiotic resistant strains is a high risk. And in the hospital, poor implementation of infection prevention, limited resources, poor awareness about healthcare professionals lead to a high burden of endemic healthcare associated infections. So in every way, we are actually increasing the burden of infections. Ingestion of contaminated meat. While some countries, for example, today Singapore used to import a lot of meat from countries like ours, but now they have stopped it because they know that in India there is no regulation on the amount of antibiotics that are used on animals. So if every country says we will stop the amount of antibiotics administered to animals or in fish culture, then there will be a huge difference, because when we ingest those animals, again those antibiotics remain in our system and we develop resistance, and so our whole system is getting changed because of the presence of antibiotics in all these things. Then contaminated water. It is estimated that 35% of the people in the region are exposed to feces contaminated drinking water, right? And look at the lower part of the same graph. In animals also, there is a huge amount of use, and few countries, there are only few countries which have legal framework for antibiotic stewardship in animals. Environment, apart from water, antibiotic residues are released into the environment from human activities, effluents from healthcare sites, inappropriate disposal of unused antibiotics, and pharmacological waste pose a significant risk. So, if you look at the scenario, you feel no doubt why India has become the capital of antimicrobial resistance, because we have all the problems that have been listed in this infographic. So it's very easily available. It's free online, right? So what is the other difficulty that we face? The other difficulty that we face in our country is we have a large burden of infections. They are often fatal. There's limited access to drugs by a large section of population. So how are abscesses dealt with? Classically, किसी के पास भी जाके कटवा लेते हैं. They don't bother कि, I mean, I won't say they don't bother. I would say they do not have access because of poverty, because of ignorance, because of unavailability of medical facilities. Sitting here in Bhubaneswar in the audience, if you cannot talk of something like Karwa art, how can you expect even basic culture facilities available in the rural periphery where you get serious infections? So we have a huge country, a huge burden of infections. Many of these are fatal, and so actually the government should liberalize access to antimicrobials because we have so much infection. Many of them are fatal. Patients don't have access. So antibiotics, which are the top end, should be given free of cost. But does it always work? On the other hand, we have the other situation where we have large pool of resistant organisms, highest levels of antimicrobial resistance, and so the government should restrict access to antimicrobials. So two contradicting situations going simultaneously. Maybe one is a little more in the remoter areas, one is a little more in the city areas, but that's not completely true. You have a huge slum population in every city, and they would be mostly very poor, living in closed areas with rapid transmission of serious infections. So that is what happens. That's why we get epidemics. So this is a major problem. Government is putting so much of energy into influenza, giving free of cost Tamiflu, doing the H1N1 testing free of cost, whereas people are dying of simple things like dengue and typhoid and abscesses, serious staph infections. So it's really not easy. We have to deal with a lot of things. So what are the solutions? Who are the ones who all need to wake up and say, we take this on as a big challenge and India is going to bring down its infection rate and its resistance rate? The clinician needs to act. We will hear more about it. The microbiologist has a big role to play. We cannot say that we close our eyes to the Department of Microbiology. They just give us some stupid reports and we are not going to look at them. We know everything about antibiotics. That cannot happen. They have to go hand in hand, and rounds by microbiologists have to be insisted upon, and regular discussions with microbiologists are necessary. I think we as clinicians tend to look down upon all non-clinical branches and think we are superior to them, we understand everything better than them. उनको क्या है, वो तो लैब में बैठ के सिर्फ बैक्टीरिया देखते हैं, हमें तो मरीज देखना है, हमें तो मरीज हमें पिटाई करेंगे अगर मर जाएगा. That is not completely true. Take them on board, like she said, take them out for a tea or something, maybe give them presents, give them gifts on their birthdays, take them along with some clinico-microbiological correlation should be. Hospital administration plays a very important role. Implementation of infection control policies and waste management. Don't forget the way you as a clinician discard your infectious material has a big role to play in the continuous propagation of these infectious things. Pharmacologists and drug manufacturers, policymakers, we think they have no role to play in how you and I treat patients, but unfortunately that doesn't work that way. Infection prevention at every level, just like I really loved Sunil's last slide, hand hygiene, hand hygiene, hand hygiene, cannot be stressed more than that. So, at the community level, sanitation and clean water, a big dream for India even today after so many years of independence, it's not happening. So actually, PM Modi's call for Swachh Bharat is a and I think doctors should embrace it the most, because if you can really improve the situation in the community, your pool of resistant organisms is going to go down. Vaccination is very important. If you can vaccinate people, children, they are not going to have XYZ diseases, and they don't need to come to the hospital. They are not going to get those infections that are going to spread further resistance. Hospitals and healthcare organizations need to be strict. Disinfection, sterilization, hand hygiene, isolation of infected patients, antimicrobial stewardship, and of course waste management, all are equally important. Unfortunately, these are not given their due importance. There will be a lot of things said about it, but nothing really followed, and nobody to check whether you are actually implementing it or not. Okay, how can you improve the life of current antibiotics? Cycling, mixing, using shorter durations, and combination therapy. Let's look at each of them. What is cycling? What is cycling of antibiotics? You use one group for some time for all patients, then you stop using that group, and then you move on to another one. In theory, it seems very good, but the thing is, when you remove that one group, the resistance to that group doesn't go off immediately. It's not like an on-off switch that you turned off that antibiotic and the resistant bugs to that particular antibiotic completely disappear. It doesn't work, unfortunately, that way. Moreover, now bacteria are much smarter than us. So if they have resistance to A group, they also may have resistance to B group. So by changing from A to B, you did not achieve much. Right? Mixing means you want to confuse the bacteria. If you have 10 patients, you give them 5 different classes of antibiotics and see to it that that 2 patients next to each other do not get the same group. But again, it doesn't work. So unfortunately, all these things were talked about a lot. Repeated studies have shown that they don't work to that extent. They may still work if your baseline level of resistance is very low, but we know that especially in our ICUs and our hospitals, the level of resistance is pretty high, so they do not work. Using shorter durations of antibiotics, as I pointed out in my first lecture, duration is something that we all must be very cautious about. Too short is also bad, too long is also bad. Know your pharmacology, know your pharmacokinetics. Something like azithromycin for 3 days is supposed to be a good course, whereas if you are using something else, you may need a 7-day therapy for a serious lower respiratory tract infection. Combination therapy is something that we are using in, which are the diseases in which we are using combination in the community? Yes? Tuberculosis, one more disease? HIV, right? So this is something approved so that you are kind of, if there are different mechanisms of resistance, you are ensuring that you are not allowing the bug to get resistant by giving him a combination from different groups of antimicrobials. But does it work in our other situations? So we say, I'm going to combine one for gram positive, one for gram negative, one for anaerobes, and one for antifungal, fungus. Ideally, that's not correct. And once you know what you have got, you must try to stop it. And nowadays, most of the drugs that we are using, except vancomycin, are all broad spectrum, like your meropenem, imipenem, tigecycline. All these have a very broad range of antimicrobial spectrum. So combination therapy is useful, but it has its own limitations and you can't use it injudiciously. The other strategies are assure and ensure drug levels. The microbiologist spoke about therapeutic drug monitoring, but again, it is not available in most of the centers. The role of generics, I am very wary of generics. I agree that the first imipenem that came may be the costliest drug, and I can get the same drug at some 200 rupees, whereas the original molecule costs something like 2000 rupees. But if you are really treating a serious infection in a salvageable patient, maybe you should not go for generics. Unfortunately, the most salvageable patients are often the poorest. So you end up using generics in those patients, and it will be those hi-fi, useless, 80 plus year old patient in whom you will be using the costliest drugs because they can afford it. Well, that is the reality in India. Using older drugs which are not frequently used or withdrawn, colistin has come back in such a big way. Maybe the next big drug would be chloramphenicol. Already some people are using it, but we should be aware that they all have their toxicities. I would just like to say that amphotericin B is not so bad. What we need to know is that you need to give a prolonged infusion. If you can give amphotericin over 10 hours, 8 hours, or at least 6 hours, it is not so nephrotoxic. Ultimately, the toxicity comes from the cumulative dose, right? So if you give it for 2 weeks, 3 weeks, it's not that toxic. It is only when you were using it for things like Kalazar, where you needed very extended spectrum, that you got it, and take care of the other things, the hydration of the patient and the other nephrotoxic drugs. Just a word about outpatient parenteral antibiotic therapy. So we know that hospitals are a closed unit, and there's a lot of transmission of resistant bugs. Anybody who sits in a hospital, a patient who's on a hospital bed for even 24 hours gets colonized with a lot of resistant bugs. So discharge patients as soon as possible. Continue parenteral antibiotics on an outpatient basis. Once you are sure the patient is going to be stable and at home. I don't know about other centers, but as pediatricians, we often use it. Like a neonate with suspected sepsis who needs a 7-day therapy, and the baby is otherwise doing well on full feeds, 2 days or 3 days later, you say, okay, go home, take these antibiotics locally, you put an IV line and send him home and say twice daily or thrice daily. It is a pain. The parent has to take the baby twice or thrice a day to the nearby center and give the antibiotic, but it is better than the patient sitting in the hospital, occupying a bed, increasing the cost, and taking on resistant bugs. And of course, for the relatives, it is so much easier. They can go back home. Home environment is always liked. So decide on dose, route, duration before discharge, and of course, there has to be a system, in which case there is a problem, they're going to report to you. And what is hospital antimicrobial stewardship? It's an interprofessional effort across all departments, which looks for best patient outcome with least toxicity and minimal impact on resistance and the environment. It is not just antibiotic policy. That is completely different from a hospital antimicrobial stewardship program, because it takes on the microbiologist, the ID consultant, the clinician, all are taken on. And there is a person who is from the administration who ensures that what is being said is being followed. At least it doesn't happen in SDPGI. I'm not sure where all it happens. Now, the role of rapid infection diagnostics, can't overemphasize the role. These are rapid and reliable methods which can help the clinician in streamlining therapy. Secondary and tertiary level, we've already heard about it. Primary level, at least we should have inflammatory marker assays, and WHO is trying to involve a lot of cheap tests with minimal infrastructure, like maybe chromogenic tests, which can be done easily, like the color changes, so you know that, okay, this is this. But as I said, many clinicians are not wanting to even go for a diagnosis, but I think that's not right. Even if it's a OPD patient who's coming for a UTI, we should encourage clinicians to go ahead to do a urine routine and a urine culture. It doesn't cost much, it will give a good sensitivity. You may actually be able to narrow the spectrum, and the patient can probably be spared the cost of a much costlier antibiotic. Such things can be easily done. And very big role to be played by the pharma industry, but I think they are more interested in making money, ensure proper drug quality, promote research on new drugs. There are countries which are now actually giving huge grants to the pharma companies to come up with newer antibiotics. But as I said, antibiotics have a lifespan, so they are not interested in antibiotics. They are more interested in lifestyle modifying drugs like anti-diabetics, antihypertensives, anti-obesity drugs, and so on and so forth. We need people who will promote the development of reliable and rapid diagnostics and promote the development of newer and effective vaccines. The rotavirus vaccine can actually revolutionize pediatric outpatient management, because if diarrhea stops, then it's a big achievement. If respiratory tract infections, they cannot stop because you cannot have vaccines against every virus, but you could have a large part of the serious LRTIs taken care of by the H influenza and the pneumococcal vaccine. So it's vaccines have no role to play. This is where I think we really struggle. Environmental sanitation, clean drinking water, vaccine, regulation of available antibiotics, available drugs, national antimicrobial policy, and encourage and fund research. So this is completely not in our hands, but people who are at the top can always have dialogues with the government. Like, for example, the Indian Academy of Pediatrics constantly guides the national immunization policy. So at least that part is taken care of. The implementation of the vaccination is obviously not in the hands completely of the pediatrician, because when it goes into the periphery, it is by the health workers. So that cannot be ensured, but at least we can guide what is being done, like this antibiotic policy. If some stakeholders take hold of it and say that this has to be strictly implemented, then perhaps it will work. The most important is the One Health approach, integrating the health needs of humans and animals, stop antibiotics to animals for growth and as prophylaxis. The hygienic standards that are required to rear animals are not maintained. So as a result of that, prophylactic antibiotics are used so that a large number of the livestock don't die. But that is not the way it should be. Rather, it should be like promote the hygiene of the while rearing the livestock. For example, if you go to poultry farms, you see how much of filth is there. The whole place is stinking. Why can't it be ensured that that place is looked after properly, rather than giving prophylactic antibiotics to all the chickens? So minimize use of antimicrobials in agriculture and aquaculture. So again, in the fish industry, a lot of antibiotics are used. So what does the future hold? It seems that everything is so bad. We have no future. 2050 is doomsday, and we're all going to die of simple silly infection. One boil and you're killed, right? One tooth extraction and you're dead. So all doctors are going to be out of jobs because antibiotics are not going to work. It's not like that. There are certain novel targets and novel strategies, and what you can utilize are what is already available in nature. So normally bacteria have a toxin or a toxic substance, but if that toxic substance is lying free within the bacteria, it will destroy the bacteria, right? So nature has made it such that it is enveloped all around by an antitoxin. And what does this antitoxin do? It protects it till it comes to a place where it is to be released, and then only it is released. So these antitoxins, if we can harness their energy and ensure that they are annulled, then the toxin is released, the bacteria die themselves, and they cannot kill you and me. They will have their own, they will lead to their own death. So this is a strategy that is being used, and this is already present in nature, and we just need to learn more and more and find out ways of harnessing this. As we have heard, most of the resistance is carried in genetic elements which are mobile, which can move from one bacteria to the other, same bacterial species and across species also. So, if you can destabilize these mobile genetic elements, it would be wonderful. A lot of substances are secreted. So if we can inhibit all these protein synthesis, nucleic acid synthesis, cell wall synthesis, transcription and translation. So at each of these steps, there are substances which are secreted. Inhibition of protein-protein interaction, like this toxin, antitoxin, both these are basically proteins. Then there are certain things known as fragments. So now drug discovery is moving on to discovery of fragments. You have these very small particles which you then try to transmit on to the patient, and then they would do their work by targeting the bacteria and sparing the humans. And of course, antisense RNA. Another thing is the biofilm. We'll just look at it. So what is it that is actually in research? This is a 2016 article from which I have taken. Some of them are in phase 2 trials, some of them are in phase 1 trials, using bacteriophages. We know that bacteriophage infects, a cell. So these bacteriophages can be incorporated with a genetic material which you have engineered and let them go and kill all the bacteria. How to give it is the biggest problem, because do you target it within that organ? Do you give it into the blood? That is the problem. Using enzymes, lysins, which break cell wall. We know that bacteriophages, when they infect the bacteria, they break the cell wall. So they use lysins. So harness those lysins and try to use them. Inhibit quorum sensing, we'll see that. And these are targeting the nucleic acid synthesis, topoisomerases, target antimicrobial peptides, and come up with efflux pump inhibitors. We were hearing a lot about efflux pumps. So there's a huge list of some 19 substances which are being tried, but don't know whether they will work or not, just like many antibiotics come and go. So quorum sensing is again something related to biofilms. Microbes communicate with each other and exchange signaling chemicals. These chemicals act like autoinducers. They allow a bacterial population to coordinate gene expression for virulence, conjugation, apoptosis, mobility, and resistance. So a whole colony of bacteria comes together with a collagen base. It forms a film. Your ETT, your central line, everything has a biofilm, especially, and your implants. Now these happen because of chemicals which are being exchanged, and these signaling chemicals can be targeted, and you can actually try and give inhibitors of these. So it's a very potent method of inhibiting bacterial virulence, and some have been found to be naturally occurring, example in garlic extract, and some have been synthesized, and there are many animal model studies which have shown promising results. So it's not that we do not have any armamentarium in the future, it's just that perhaps we have come up with a huge amount of data. We don't know what to do with it, but we will probably learn slowly. Another aspect which I have not mentioned, just like we have Clostridium difficile, one of the last modalities of therapy is fecal transplant, right? So you take feces from a normal individual who will have healthy bacteria, and you infuse it into the person who's infected, and you hope that the C. diff is repressed, and you have the normal healthy flora take over, colonize the gut, and get back. So the role of the gut microbiome is very important, and perhaps in sepsis patients also we can do something similar. It sounds very scary that you're actually feeding fecal matter to a patient from someone else, but that is one of the things that will perhaps, ultimately probiotics, that is what we are trying to do. We are trying to feed them good bacteria and hoping that those good bacteria will colonize the gut. Like our old grandmas used to say, diarrhea hai, curd khilao, theek ho jayega, right? So basically curd is full of probiotics. So these are all the things that I really love. The Lancet Infectious Diseases Journal, one of them carried, the lead article was on naturally occurring antibacterial substances available in the marine flora, and this was the May 2016. UK came up with this report by an economist. So an economist was asked, how do you tackle this problem of drug-resistant infections globally? And CDC's Center of CDDEP, Center for Disease Dynamics, Economics and Policy, Ramanan Lakshminarayanan, I really admire this man. He writes wonderfully. The first few lines in my first lecture was from his editorial. He's the director of CDDEP, and CDC is so worried, the world is so worried that one of the centers of this, he's the director in India, in New Delhi. So basically, because India is the capital of drug resistance, unfortunately it seems the government is more interested in regulating how cows are looked after rather than trying to regulate infectious diseases. So maybe if cows are looked after better, even humans get better, who knows. So I leave with that note. Antimicrobial resistance is playing havoc in medicine as part of the medical community. We have a responsibility. I would like to leave this thought with you that we have a duty. It is not just that, something which you need, which you can do, you should do, we all need to do, to ensure that we don't go back to the pre-antibiotic era. And it's a One Health approach that is probably required urgently, and nothing is more true than this poem by Robert Frost. The woods are lovely, dark and deep, but I have promises to give and miles to go before I sleep. Thank you.